So this presentation is on identification of carbohydrate, particularly fructose. Coming to the physical properties, state is liquid, the solution is colorless, odorless, and the reaction to litmus is neutral. So these are the different tests we perform to identify presence of fructose in the given solution. So these are the explanation of the different tests. Coming to the first test, Mollis test, general test for carbohydrates, we get to see formation of violet coloring at the junction of the two liquids. So what's the principle behind this test? Carbohydrates, when treated with concentrated sulfuric acid, undergo dehydration to form furfural derivatives, and these furfural derivatives in turn condense with alpha naphthol to form a violet colored complex. Next is iodine test. So there will be no color change, which indicates polysaccharides are absent. So the solution may be either mono or disaccharide. The next test is Benedict's test, which indicates formation of different colored precipitate. Here it is orange colored precipitate. So different color indicates different concentration of the sugar present in the sample. Green indicates 0.5%, yellow 1%, orange 1.5%, and red indicates 2%. Now what's the principle behind reducing test? Now this test is mainly done to identify the presence of reducing sugars in the lab. So reducing sugars in hot alkaline medium are tautomerized to form enediols, which are powerful reducing agents. They reduce the copper sulfate of Benedict's reagent to cuprous oxide, and on boiling, so this cuprous oxide gets converted to green, yellow, orange or red colored precipitate. So this is reason why this Benedict's test is known as both qualitative test based on the color development, and based on the constant different color we can it indicates percentage of different sugar present in the precipitate. So it is both qualitative and semi-quantitative. Now we got to know that the solution is carbohydrate, it's not a polysaccharide, it is reducing in nature. So now next test is we have to know whether the solution is a monosaccharide or disaccharide. So we now perform Barfoot's test. So we get to see red granular precipitate at the bottom of the test tube. So this indicates the presence of monosaccharides. Now what's the principle behind this test? Monosaccharides with free aldehyde or keto group can reduce cupric ions to cuprous oxide in acidic medium. So the difference between Benedict's and Barfoot's is Benedict's is test is done in alkaline medium, whereas Barfoot's test, the test is done in acidic conditions. Now we get to know that it is a monosaccharide. So next identification is to know whether this monosaccharide is a aldo sugar or a keto sugar. So we need to perform Selivanov's test. So we get to see cherry red color here. This is a light cherry red color. Now this indicates the presence of ketose, that is fructose. So what's the principle behind this test? Resorcinol, which is present in Selivanov's reagent, react with furfural derivative to form a cherry red colored complex. So how to report the sample? So the given solution is carbohydrate reducing monosaccharide keto sugar, and thus ketose is fructose. Coming to sources, properties, functions and clinical significance of fructose. As you all know, fructose is mainly present in fruits, honey, and it is one of the component of sucrose, which is disaccharide made up of glucose and fructose. Properties of fructose are as follows. It is a keto sugar. It has got a functional keto group present in furanose form, and it is levorotatory, means it rotates the plane polarized light to the left side. So fructose is reduced to mannitol, its alcohol form, and when fructose is made to react with phenylhydrazine, it forms broomstick shaped ozone crystals. So fructose can also be converted to amino sugar. It's reducing in nature because it reduces the cuprous ions of Benedict reagent to cupric ions. Now fructose is also present in inulin, which is a homopolysaccharide of fructose. Coming to functions of fructose, it provides energy for the motility of the spermatozoa. So next important function is it is more rapidly utilized than glucose. Now this is the pathway of glycolysis, which starts with phosphorylation of glucose to glucose 6-phosphate, which is isomerized to fructose 6-phosphate and gets converted to fructose 1,6-bisphosphate, glyceraldehyde, and this is broken down to glyceraldehyde 3-phosphate and dihydroxyuracil phosphate, and so on. At the end it gets converted to pyruvate. So now just see how fructose is metabolized in the body. Fructose gets phosphorylated to fructose 1-phosphate. Now this is acted upon by fructose is getting converted to fructose 1-phosphate by fructokinase enzyme, and fructose 1-phosphate is broken down to glyceraldehyde 3-phosphate and dihydroxyuracil phosphate by aldolase B enzyme. So the subsequent steps are similar to that of glycolytic. What we need to observe is the first two steps of glycolytic, when glucose enters the glycolytic pathway, is bypassed by the fructose. That's the reason why fructose is more rapidly metabolized than glucose. So coming to the clinical significance, when fructokinase enzyme is deficient, which is involved in converting fructose to fructose 1-phosphate, it results in an inborn end of metabolism called fructosuria. When aldolase B enzyme is deficient, which is involved in breaking down fructose 1-phosphate to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, it results in a condition called hereditary fructose intolerance. The other clinical significance is mannitol, which is formed from reduction of fructose, is used in the treatment of intracranial pressure, where it relieves the water content from the cerebral part of the brain by diuretic effect. So this was all about clinical significance, functions and properties fructose. Now this presentation was done by Dr. Preeti, Professor of Biochemistry from JJ Medical College, Davangere. Now here are the links to other practical biochemistry part. You can access the different practical aspects of biochemistry by clicking on these links. Thank you.